Good day, all. Welcome again to another edition of Dr. Ify's Hematology and Blood Transfusion Science Lecture Series. Today, we are going to be discussing another blood group system, MNS Blood Group System. It is expected that at the end of this lecture, you should be able to know the antigens in MNS Blood Group System, should be able to describe the antibodies in MNS blood group system, and also understand the genetics and inheritance of MNS antigen. And of course, should be able to explain the clinical importance of MNS antibodies. History. MNS was the second blood group discovered in 1927 after the discovery of the first blood group, the ABO blood group system, in 1900. Landsteiner and his colleagues discovered this after immunizing rabbits with human red blood cells. The M and N antigens were identified first, but it was another 20 years before the S and the little s antigens were named. The MN and S little s alleles are linked on chromosome 4. Therefore, they belong to a single blood group system. The antigens in this blood group system, how many are there? We have about 43 antigens in this blood group system. But the prominent ones are the M, N, big S, and little s antigens. The ISBT symbol for these antigens in this blood group system is MNS, upper letter M, upper letter N, and upper letter S. And then the ISBT number is 02. The nature of antigens in this blood group system. The antigens are proteins, and amino acid sequence determines the specificity of the MNS antigens. The antigens of the MNS blood group are carried on sugar-bearing proteins called glycophorins. These glycophorins lie in the red blood cell membrane. One end of a glycophorin is attached to the underlying cell, and on the other end bears the sugars and determines the person's MNS blood group. The glycophorins A and B are single-pass transmembrane proteins. Glycophorin A contains abundant sialic acid, which contributes to the negative surface charge of the red blood cell membrane. It has three main domains: an extracellular domain having 70 amino acid, the membrane-spanning domain having 22 amino acid, and the intracellular domain having 39 amino acid. The M and N phenotype differ from each other by one amino acid at position 1 and 5 in the extracellular N-terminal domain. The glycophorin B is structurally similar to glycophorin A, also consisting of 3 domains, but with a shorter intracellular domain of 6 amino acid. The big S and little s phenotypes differ from each other by 1 amino acid at position 29. MNS antigens. The M and N antigens differ by 2 amino acids. Glycoprotein antigens are destroyed. These antigens are destroyed by enzyme treatment. Very, very important. They are destroyed by enzyme treatment, just like antigens in Dufy blood group system. you need to know the antigens that are affected by enzyme treatment because it will help you in identifying their antibodies when you are doing antibody identification. So the antigens in MNS blood group system are destroyed by enzyme treatment, just like that in Dufy blood group system. They also have dosage effect. Also very important because of they are needed in homozygous form when you are preparing your selectogen, your panels for either antibody screening or antibody identification. They have dosage effect, that is quantitative variance, just like the antigens in kid blood group system. The S and little s antigens differ by a change in one amino acid. And the big S is more commonly seen in those who are antigen M positive than those who are antigen M negative. Then, function of the molecule that carry the MNS glycoprotein. The molecules that carry the MNS glycoproteins is like the glycophorin A and B. What do? Their functions. Glycophorins A and B may serve as receptors for cytokines, bacteria, and viruses. But the lack of the glycophorins does not result in any form of disease. Those that don't have them, they don't suffer from any known form of disease, indicating that their function is not physiologically significant, at least in modern times. Scientists are also interested in these glycophorins because they bear the MNS antigens and because they may act as a receptor for Plasmodium falciparum. This parasite is one of the parasites that cause malaria infection in humans. Individuals who have rare blood type, that's people that lack glycophorin A and B, the research has shown that this set of individuals are resistant to invasion by Plasmodium falciparum. Expression of MNS antigens. The MNS antigens are found mainly on red blood cell membrane. There are about 1 million copies of glycophorin A per red blood cell membrane and 0.2 million copies of glycophorin B. The MNS antigens are also expressed in the kidney, on the renal endothelium, and the epithelium. Frequency of MNS antigens. The M antigen can be found 78% in Caucasians and 74% in blacks. The N antigen can be found 72% in Caucasian, 75% in blacks. The big S antigens are approximately 55% in Caucasians, 31% in blacks, and the little s antigen is about 89% in Caucasians and 93% in blacks. Now the phenotype, the genotype, and the approximate frequency in white and blacks, and then reactivity with anti-M and anti-N. For those who is having N plus N minus, those individuals, they have the M antigen, they don't have the N antigen. So their genotype is MM. They are about 28% in white, 26% in blacks, and then because they have the M antigen, they react positively with anti-M and react negatively with anti-N. Those individuals that are M plus, N plus, they have the genotype MN, and you have about 50% of them in whites, 44% in blacks, and because they have both M and N antigen, they react positively to anti-M and positively to anti-N. You have those that are negative for M but positive for N, having the NN genotype. And they are about 22% in whites, 30% in blacks. Because they don't have M antigen, they react negatively with anti-M and positively with anti-N. Now, with little s phenotype, those that are positive for big S and negative for little s, they have their genotype as big S, big S. You have approximately 11% in whites, 3% in blacks, and because they have big S, they will react positively with anti-S, anti-big S, and negatively with anti-small s. Those that have big S positive and small s positive, that is, they have big S antigen and small s antigen. Their genotype is big S, small s. And you have about 44% of these individuals in whites, 28% in blacks, and because they have both the big S and small s antigen, they react positively with anti-big S and anti-little s. Those that are big S negative or small s positive, they have their genotype as small s, small s. They have their genotype as small s, and they are about 45% in whites, 69% in blacks, and react negatively with big S, but positively with small s. Common phenotypes. In Caucasian, the most common phenotypes are M plus, N plus, S plus, S plus. And you have about 24% of these individuals in Caucasians. Then M plus, N plus, big S minus, small s plus. You have about 22% in Caucasians, and M minus, N plus, big S minus, small s plus, about 15% in Caucasian. The latter two phenotypes are common in blacks also, occurring at a frequency of 33% and 19% respectively. Antibodies to MNS antigens. The antibodies to MNS antigens are both IgG and IgM. You can have IgG, you can also have IgM. The IgG class depends upon which antigen that is targeted. Transfusion reaction, they give rise to, the transfusion reaction is actually uncommon, but potentially severe. They can cause transfusion reaction, but it's very uncommon. But when they do, it can be severe. And big S and little s are among the MNS antibodies implicated in causing transfusion reaction. They can also cause hemolytic disease of the newborn, but it is also rare. But when they do, it can be severe. And big S is more common than anti-little s, but both are capable of causing severe to fatal hemolytic disease of the newborn. Anti-M and anti-N. Normally, anti-M and anti-N are normally IgM in nature. They can have IgG component, but normally they are IgM in nature. So they are called antibodies. They can react at room temperature. Usually stimulated by transfusion, but many examples are natural occurring. Anti-M is more common in children and patients with bacterial infection. Do not readily activate complement. They don't readily activate complement. They usually react by saline technique up to 20 degrees Celsius. So you can say they are cold antibodies. Some examples of anti-M may react at 37 degrees Celsius by anti-human globulin technique. They have little clinical significance, rarely cause transfusion reaction and hemolytic disease of the newborn. Anti-big S and anti-little s. These ones are generally immune. They are mostly IgG in nature. They are rarely produced. They are normally IgG, but you can also have IgG plus M, and they are complement binding. They are clinically significant. They have been implicated in transfusion reaction, and they have been also been implicated in to cause severe to fatal hemolytic disease of the newborn. Anti-big S usually reactive by anti-human globulin technique at 37 degrees Celsius. Anti-little s may be reactive at 20 degrees Celsius, saline, as well as 37 degrees Celsius, anti-human globulin. And it can have variable results with enzyme technique. Anti-U, produced by U-negative individual, is non-complement fixing IgG antibodies and react optimally at IAT, that's indirect antiglobulin test, and implicated in transfusion reaction and hemolytic disease of the newborn. The genetics of MNS antigens. The two genes that encode the glycophorin that carry the antigens of MNS blood groups are GYPA and GYPB. Both are on the long arm of chromosome 4, and they are tightly linked together, and a combination occurs between them. A third gene, GYPE, is located next to GYPB and may play a role in the gene arrangement that results in new variant alleles. GYPA and GYPB are similar, sharing up to 97% sequence homology. In fact, 5' GYPA and GYPE prime gene cluster is thought to have originated from a single ancestral gene that underwent 2 duplications. The GYPA locus. The GYPA gene consists of 7 exons that span more than 60 kilobytes. It has 2 allelic forms called MNS1 and MNS2, which produce the M antigen and N antigen respectively. The alleles are identical except for 2 amino acid substitutions. The MNS1 alleles encodes serine at residue 1 and glycine at residue 5. The MNS2 alleles encodes leucine at residue 1 and glutamate at residue 5. The GYPB locus. The GYPB locus gene consists of 5 exons that span more than 58 kilobytes. It has 2 allelic forms called MNS3 and MNS4, which produce the big S antigen and the little s antigen respectively. They allele differ in 1 amino acid. The MNS3 allele encodes methionine at residue 29, whereas the MNS4 allele encodes the threonine at this particular position. Now, what are the clinical significance of MNS antibodies? They are capable of causing transfusion reaction. But anti-M and anti-N are not considered to be a cause of transfusion, so they are not clinically significant. Although rare cases of delayed transfusion reaction have occurred as a result of anti-M. Anti-M is very common and is thought to mostly be naturally occurring because it is frequently found in children who have never received a blood transfusion. Mild to moderate transfusion reaction can be caused by the presence of anti-big S and anti-little s in the patient serum. Severe transfusion reactions have been attributed to anti-U, anti-VW, anti-MOR, and anti-ENA. They can also cause hemolytic disease of the newborn. Of the MNS antibodies, anti-big S is more common than anti-little s, and both are capable of causing severe hemolysis. Less common causes of hemolytic disease of the newborn include anti-M, anti-N, anti-U, anti-MIA, anti-MTA, and anti-ENA. Other MNS antibodies implicated in HDN are anti-VW, anti-MOR, anti-HERT, anti-HALE, and anti-MV, anti-FAL, and SD. In addition, other antibodies to low incidence of MNS antigen should also be considered as potential harmful. I am sure that today you can give an overview of what an MNS blood group system is all about, the antigens that can be found in MNS blood group system, the antibodies, their reactivity, and their clinical significance. These are my references for further reading. Feel free to give us feedback. Thank you very much.